Hi, this is Vinit Kurnala. I'm collaborating with Megan Curley and Saba Raja for the final project of statistical analysis of genetic data. We selected a publication from International Heart Journal, which is analysis of gene expression during the development of congestive heart failure after myocardial infarction in rat models. And we selected this publication and we are trying to summarize it using different methods. So the objectives and the study design are as follows. The objectives of the study is to understand the development mechanisms of congestive heart failure after myocardial infarction at different phases at genomic level using the rat models. And the study design is we had the original publication at a total of 24 samples, 6 samples of left ventricular myocardium from male Wester rats 1 day after MI, and such samples from 1 day after sham, six such samples from 7 days after MI, and six samples after sham, 7 days. And they performed gene expression analysis using the GPL 153 Atlas Rat 1.2 array. So what is so the data that they used was the microarray data. And what is microarray data or microarray? So microarray is a technology used to monitor genome-wide expression levels of genes in a given organism. A microarray may contain thousands of spots, and each spot may contain a few million copies of identical DNA molecules that uniquely correspond to a gene, which is shown in the figure here. The DNA in a spot may either be DNA or short stretch of oligonucleotide strands that correspond to a gene. So these spots are printed on a glass slide. The spots are arranged in an orderly fashion into pen groups. Uh, and The next figure shows the schematic of the experimental protocol to study differential expressions of genes. The organism is grown in two different conditions, a reference condition and a test condition. And the RNA is extracted from the two cells and is labeled with different dyes, red and green. Following this step, the complementary DNA is hybridized onto the microarray slide, where each complementary DNA molecule represents a gene, and it will bind to the spot containing its complementary DNA sequence. The microarray slide is then uh, excited with a laser at suitable wavelengths to detect the red and green dyes. The final image is stored as a file for further analysis. And zooming onto the spot of this microarray slide, the spot area and the background area depicted by a blue circle and a white box, respectively. So a pixel in the spot area is also shown here. Any pixel within the blue circle will be treated as a signal from the spot, and any pixels outside the blue circle within the white box will be treated as a signal from the background. So we can see that the images are not perfect, but it's often the case. So this may lead to problems uh, with disturbed signals from dust particles and scratches on the glass lights and bright arrays. So the key data processing steps that are generally used in the microarray experiments are background correction, normalization, and filtering. So the background correction is nothing but removal of an ambient non-specific signal from the total intensity in the image analysis of two-color microarray experiment. So, so basically what background correction does is subtract background signal from the spot intensity to get a more accurate estimate of the signal from the spot. And then so there are different types of background corrections, and so one of them is the local subtraction. The next one is the normalization. So normalization is nothing but removal of systematic variation in a microarray experiment which affects the measured gene expression levels. So since we are dealing with the microarray data here, normally microarray data can consist a lot of systematic variation So the normalization removes this variation, and also it affects the uh, removes any systematic variation which might affect the measured gene expression levels for various reasons, such as the experimental bias, variability in experimental conditions, sample collection preparation, or the machine parameters. Um, the next step is sorry. In the normalization, in many cases, there can be differences in processing of two samples, particularly during the processes of making the cDNAs. Labeling and hybridization, as shown in the previous figure, systematically affect the signals on the arrays. Some systematic processing differences between the chips that frequently affects the measures are different amounts of RNA that are used in the experiment, or one dye is more readily incorporated than the other, and different amounts of labeling may occur in the color systems. So the next one is the filtering, which is nothing but reduction in the number of hypothesis tests performed and a corresponding increase in power. Due to the large number of hypothesis tests performed during the process of routine analysis of this microarray data, a multiple testing adjustment is certainly warranted. However, when the number of tests is very large and proportion of differentially expressed genes is relatively low, the use of multiple testing adjustment can result in very low power to detect those genes which are truly differentially expressed. So the filtering allows for a reduction in the number of tests and a corresponding increase in power. Some common filtering methods include filtering by variance, average signal, or MAS detection signal. So the data processing reported in the original publication are as follows: quantile normalization on the raw gene expression data, mapping probes to genes, and applying linear regression with procedure MI or a sham and time points as covariates. So quantile normalization is a technique for making two distributions identical in statistical properties. To quantile normalize a test distribution to a reference distribution of the same length, we sort the test distribution and sort the reference distribution. The highest entry in the test distribution then takes value of the highest entry in the reference distribution, the next highest entry in the reference distribution, and so on, until the test distribution is a perturbation of a reference distribution. The next step is to map probes to genes. So here in expression arrays, a probe is a short DNA sequence targeting a short region of a transcript. They are used to detect the presence of nucleotide sequences through hybridization to single-stranded nucleic acids due to complementarity between the probe and the target. And typically, the probes are 25 BP to 60 BP long. In the Affymetrix arrays, probes are grouped into probe sets, which are designed to target the same transcript with multiple measurements. In the original articles, probe sets were mapped to the genes. If there were more than one probe set then that corresponded to the same gene, the average value was calculated and considered as the expression value of the gene. The probe that had multiple genes was filtered. So the data processing that we did was is as follows. For the normalization, our goal was to remove batch effect, otherwise it would bias the results, and then apply quantile normalization to the raw gene expression data. And after normalization, take the log2 transformation of the normalized data, as the gene expression data is always skewed and log transform data is more normally distributed, and the estimation from linear model would be more accurate. And so these are the steps in our data processing. So in the pre-processing and log normalization and excluding missing values. So the original article applied quantile-based normalization using the FEPLM package, although it was thought that robust multi-array function of the oligo package might present a better alternative, but the lack of CEL files from the database provided us from prevented us from taking this that approach. So the robust multi-array analysis is made up of three processing steps, and these steps include background correction, quantile normalization, and summarization. The summarization portion of the robust multi-array analysis procedure is based on robust model fitting of the multi-array data. This fitting is performed using a median polish algorithm oligo. A robust multi-array analysis takes a vector of CEL files and outputs. Since the CEL files were absent, we ultimately had to use the FEPLM for the pre-processing. And then the function normalized expression set quantiles was used since the available data GSE1957 was an expression set. This function performs a quantile normalization and carries an internal procedure of background correction. Then log normalization was performed using the R code from GO to R. Log normalization was performed to obtain values that are more relevant from a biological perspective and were easily interpretable. The log transformation creates a symmetrical distribution that mimics the normal distribution. So we had to use the log normalization. Another argument that favors this transformation is the convenience of analysis and interpretation. For example, since the base of the logarithm is 2, a fourfold variation can be shown by dissecting the ratio histogram at a value of 2, since log, log 2 of the ratio is 2. Along with the normalization and log transformation, the expression set contained an excessive amount of missing values. Since missing values are carried out through computations, we had to exclude them so that meaningful calculations may be obtained from the available data. So these are few screenshots from the data processing that was done. Here, if you see the box plots prior to pre-normalization and post-normalization, the spread of individual probes are different, and the medians seem to lie um, around the value of 100. But after the normalization, we can see that all the median values are aligned, and these show the density plots of pre-normalized and post-normalized and log transformed data. So after post-normalization and log transformation, it appears to be the data appears to be smoothly overlapping. The next steps will be covered by collaborators Saba and Megan. Thank you.